So I have been talking about gastrulation. During gastrulation, three things generally happen. The first thing is that morphogenetic movements which result in gastrulation, where the blastula, which is single layered, excuse, is converted into a double layered or triple layered gastrula. Second event is gastrulation results in the formation of three germinal layers: ectoderm, mesoderm, and endoderm. And also, this is the stage in the embryonic stage where the antero-posterior axis is also developed. And hence, when we see the fate map of, when we see the fate map of a blastula, there is an ectoderm, which we call it as blastoderm, surrounding the blastocyst. So this is the blastocyst, this is blastoderm. We can compare it with ectoderm. But this is not true ectoderm. Why? Because here there are cells which can give rise only to the epidermis of the body wall, which we call it as, can anybody tell me what is this called as? This is called as somatic ectoderm.
Neuroderm.
Somatic ectoderm. And this is followed by cells which are destined to form the nervous system, and this is called as neurectoderm. So both of these, the blue colored one and green colored one, constitute the ectoderm. We can call it as presumptive ectoderm because this is not ectoderm in sense, because this is part of blastoderm. This is going to become the ectoderm of gastrula. Presumptive means it's an assumption, not a just assumption. This is going to become, its fate is said to be presumed. Hence we call it as presumptive ectoderm, which is going to form the ectoderm of the gastrula. And likewise, towards the lower side, vegetal pole, there are cells which are destined to form the alimentary canal, and we call this as presumptive endoderm. And there are cells which are destined to form one, that is mesoderm, which are present on either side of the endoderm. So these we call them as presumptive mesodermal cells. In addition to this, there is another one, that is cells which are destined to form the notochord, and hence this region is called as chorda mesoderm. Okay, this is the fate map of the blastula of frog and toad. Now what happens during the process of gastrulation? These cells exhibit morphogenetic movements. So what happens now is, first of all, the thing that happens here is invagination. Invagination of what? Invagination of presumptive endodermal cells. That is, the cells that are marked here as yellow, they undergo a process called as invagination. Literally meaning going inside. So where it goes inside? Because there is a place called as blastocyst. Why it has to go inside? Because the entire development is taking place within the boundary of the egg membranes. There is no space for the developing embryo to increase in size. So it has to, whatever the new cells that are formed due to cell division during cleavage, they have to get adjusted or accommodated within the same sphere only. So as these cells are dividing and producing new cells, These cells are dividing and producing new cells. So here, as there is no space to expand for all these dividing cells, the only space that is nothing but blastocyst. What happens? The cells which are destined to form endoderm, we call these as presumptive endodermal cell. They go inside the blastocyst. So this is the somatic ectoderm, neuroectoderm, and the next one is mesoderms on either side, followed by notochord forming cells and the endodermal cells. So these endodermal cells, they do what they do. They now go inside as the cells are increasing inside. They go inside into the blastocyst. So as they go into the blastocyst, let me color it with orange so that you can see it clearly. Now we see two layers. One is this one, the other is this one. So the outer layer is called now as ectoderm. Previously it is called as blastoderm. Now we call this as ectoderm. And the inner layer, new layer that is formed is endoderm. Because of the invagination of presumptive endodermal cells. Because of the invagination of presumptive endodermal cells, the blastocyst is obliterated. That means it gets reduced. Previously, this entire thing is the blastocyst. Now it is restricted to only this region. We say it is obliterated. And a new cavity appears. And this new cavity is called by scientists as archenteron. Okay, and this archenteron opens by an opening to outside, and this we call it as blastopore. Okay, yesterday some of you did not hear this properly due to network issues, hence I again started this. The archenteron, the name itself says, arc means primitive, enteron means gut cavity. So thus archenteron refers to gut, primitive gut. So gut means we all have alimentary canal which starts with mouth and ends with anus. So if this is going to become primitive gut, so this cavity forms the gut cavity, and this wall of the archenteron forms the wall of the alimentary canal. So thus we say endoderm gives rise to alimentary canal. The cavity, the archenteron, gives rise to the gut cavity. If that is so, the gut cavity opens on both the sides, through anus and through mouth. So here, this gut cavity, primitive gut cavity in archenteron opens only on one side, and this is called as blastopore. We know already in protostomies it becomes mouth, and deuterostomies it becomes anus. So if it is deuterostomians like us, this becomes anus, so that on this side mouth develops. Thus, we during embryonic development exhibit an antero-posterior axis. So this is the stage which is responsible for the development of the antero-posterior axis during embryonic stage itself. Now we have only two germinal layers being formed now, isn't it? One is ectoderm is formed and endoderm is formed. What about the mesoderm? And also we have what we call nervous system to be formed. We have notochord to be formed, and all these things have to happen, isn't it? So different morphogenetic movements result in these things to happen. So how this takes place? Let me draw the same thing again. So this is neuroectoderm, sorry, somatic ectoderm. This is neuroectoderm, followed by mesodermal cells flanking on either side, and endodermal cells which went inside, and mesodermal cells here. Now what happens is the second step. We have seen invagination of presumptive endoderm being taking place. Now the second step is epiboly of presumptive ectoderm. Here what happens? These cells, what are these cells? Cells of the ectoderm. The cells of the ectoderm, they continue to divide after the event of the cleavage process until gastrulation is completed. The division continues. So during blastula, during morylation, during blastulation, and even during gastrulation, the cell number increases. So as the cell number increases, these cells overgrow over the other cells. They try to overgrow the other cells. In so doing, as these are moving in this direction, what happens? These cells, that is mesodermal and notochordal cells, they go inside. They go inside. So as they go inside, the ectodermal cells almost cover the entire thing. And this type of movement where the ectodermal cells, okay, they expand onto another layer is called as epiboly. So the second one is, the spelling is incorrect. Epiboly, epiboly, epiboly of presumptive ectodermal cells takes place. Okay, as these ectodermal cells are going inside, we have what we call the neuroectodermal cells here. An antero-posterior axis is developed. Now we have to develop a dorsoventral axis. What happens is, as epiboly is taking place, the the neuroectodermal cells here, they go inside. They go inside, and they form into a neural tube that gives rise to a nervous system later. So after going inside, both of these fuse and the neural tube gets separated. Okay, so now what happens? It goes inside, and after that both of these regions fuse, and we come across with a neural tube. In doing so, we come across also with certain other cells. What are they? Let us see. Go back to again. This is the formation of neural tube, where these two regions, they go inside, and these are the regions. This is the region where certain cells are present. These are called as neural crest cells. They are called as neural crest cells. So in this region, it fuses, and the neural tube is separated. So the neural tube is separated. This is the neural tube. And the neural tube expands in antero-posterior direction, leading to the formation of brain towards the anterior end and spinal cord towards the posterior end. Thus, This part, that is neuroectoderm, gives rise to the nervous system. So as these things are happening, the third event also takes place. That is, some of this, as the epiboly of these cells is taking place, the mesodermal cells and notochordal cells also go inside. Okay, so what happens now? First, these are the mesodermal. So these are the mesodermal. Sorry, this is mesoderm and this is notochord. Okay, the mesodermal cells go inside. Okay, so they go inside. So this occupies the inside of the what we call gastrula, and where they form the roof of the archenteron. మళ్ళీ పోయిందా? Okay, they form the roof of the archenteron. At the same time, the cells which are destined to form notochord also come to occupy a position at the roof of the archenteron. So these are present on the roof of the archenteron, and soon the ectodermal cells separate. Sorry. So this is the endoderm, and now the notochordal cells get separated from this. This becomes the somatic ectoderm, and we come across with mesoderm here. This is how it happens. It looks like. Okay. So now soon the notochordal cells get separated and they come to occupy a position beneath the nerve cord. Okay. So above which there is nerve cord and below which there is notochord. Even in our body plan, we have the same thing. When we see our body plan, okay, this is the ventral side, starts with mouth and ends with anus. We have alimentary canal, above which there is notochord and above which there is nerve cord. So keeping in view the body plan of the adult vertebrate, the morphogenetic movements also take place in the same way. The morphogenetic movements also take place in the same way. So thus, what is established till now? Number one, presumptive ectodermal cells got exhibited epibolic movement and ectoderm is formed. Number two, presumptive endodermal cells exhibited invagination and endoderm is formed. And thirdly, presumptive chorda mesodermal cells, they undergone involution leading to the formation of notochord, and eventually after that mesoderm is formed. Now we have to see how the mesoderm is formed. Okay, so mesodermal cells occupy a position here on the roof of the archenteron. Now we know intracellular. I am removing all this to understand this mechanism. So in intracellular, what happens? The roof of the archenteron forms into pouches. So it formed into pouches. So this is the endoderm, and pouches of archenteron with mesodermal layer formed. And soon the pouches separate by fusing. So the endoderm gets completely separated from the mesoderm, and now we have mesodermal or pouches of archenteron. which expand on both the sides, on meeting with each other, they form into two mesodermal layers. One mesodermal layer is present towards the ectoderm, that is called as somatic ectoderm. The other mesodermal layer is present towards the endoderm, that is called as splanchnic mesoderm. Thus, at the end of gastrulation, we see an ectoderm, a neural tube, a notochord, an endoderm, and a mesoderm with two layers. One towards the ectoderm, the other towards the endoderm. And this is called as somatic mesoderm, and this is called as splanchnic mesoderm. Somatic mesoderm is associated with ectoderm. Remember that. So the somatic mesoderm plus ectoderm, it is called by scientists as somatoclure. Okay, and splanchnic mesoderm. This is second layer towards the inside. Splanchnic mesoderm plus endoderm. This forms the splanchnoplure. This forms the splanchnoplure. And this is how the gastrulation process in case of amphioxus and also the frog is completed by three processes. One is epiboly of presumptive ectoderm, invagination of presumptive endoderm, and involution of cauda mesoderm. These three things you have to remember in view of the examination point of view. Now, coming to the gastrulation in polylethal eggs, we are not concerned with as per your syllabus, but just I want to mention something here. We know these eggs are said to be having yolk all over. Only this animal pole region is yolk free, where active cytoplasm and nucleus is present. So here only cleavage takes place, forming into what we call discoblastula, where only cells are present here in multi-layered cells in the form of a disc-like structure. Hence we call it as discoblastula. So when this discoblastula is seen from the surface view, we see different locations in the epiblastic region. We have discussed it in the external class. The chick embryo, during gastrulation shows epiblast followed by blastocele followed by primary hypoblast followed by subgerminal cavity and yolk material. Epiblast, and this is yolk. Epiblast is followed by primary hypoblast, and between these two there is blastocele, and between these two there is subgerminal cavity, and this is yolk, and this forms the endoderm. And now the epiblast region, this is distinguished into different regions. So when we see the epiblast, if this is the epiblast surface view, scientists say that there appears the movement of cells in the epiblast, okay, which shows a structure like this, where the cells move in the center from the center of the epiblast towards one end, and this is called by scientists as primitive streak. Okay, so primitive streak formation in the epiblast starts gastrulation in case of chick, because we are both birds and mammals are derived from reptiles, and they also exhibit the same thing, reptiles, and we also exhibit a primitive streak formation. Okay, and the other thing, so many things happen during the entire process. I'm just going to summarize this. In gastrulation, it leads to the formation of ultimately ectoderm, mesoderm, and endoderm. And these are again distinguished. into different layers. So if this is the epiblast region, okay, no, 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 I'll draw something else. Uh, this is the epiblast region, okay. These are the cells of the epiblast, okay. Now we see the hypoblast, below which there is yolk material. We can call this as endoderm, okay. Now develops what we call the mesoderm. Now develops the mesodermal layer here. So the mesoderm, when it develops, it forms different regions. And we also have seen the development of neural tube here, and below this there is a notochord. And now there is an ectoderm, an endoderm, a neural tube, and a mesoderm. Now we have to see the neural tube and notochord, and now we have to see the mesodermal regions. So here forms a mesoderm. Here forms a mesoderm, which is distinguished into different regions. This is how the mesoderm appears. This is ectoderm, this is endoderm, and this blue colored one is mesoderm. You know, mesoderm has two layers. One's towards the ectoderm, that is somatic mesoderm,
Somatic.
endoderm, that is plasmic mesoderm. And these were given different names also. The one that is somewhat bulging like this thing, okay. So this one is called by scientists. This part of the mesoderm is called by scientists. Sorry, this is called as epimere. This is called as epimere, and the one below becomes the hypomere. The one below becomes the hypomere, and the one between these two becomes the mesomere. So thus mesoderm is again divided into three: epimere, hypomere, and mesomere. And epimere is also called as somite mesoderm. It is called as somite mesoderm because it gives rise to the segmentation of the body, that is somites. Okay, and different parts of the body develop from all these layers. One is ectoderm, which is distinguished into somatic ectoderm and eurectoderm. The third one is endoderm, and fourth one is mesoderm, comprising of epimere, mesomere, and hypomere. And epimere is again subdivided into dermatome, sclerotome, and myotome. And now we are going to see what is the fate of all these different parts of the body. Fate of germinal layers. So we see ectoderm. In ectoderm, we come across with epidermal ectoderm, which is also called a somatic ectoderm, and the one which forms the nervous tissue, that is neuroectoderm, nervous system. And as during the formation of neural tube, we see certain cells here. We call them as neural crest cells. So what is the fate of these three? The epidermal ectoderm gives rise to, the first thing is epidermis of skin. So when you pick the skin and pull it, the one that is pulled out is what we call epidermis in general is. Okay. So epidermal ectoderm gives rise to epidermis. The skin has epidermis plus dermis, isn't it? Epidermis is derived from ectoderm and dermis is derived from somatic mesoderm. Somatic mesoderm. That's what you have to remember. So now the first thing that is found here is epidermis. So ectoderm gave rise to outer body wall, the outer layer of the body wall, that is epidermis. And outer layer of the body wall, that is skin, has so many derivatives. For example, human beings, primates are characterized by hairs. Birds are characterized by feathers. And the hands and feet have nails, claws. And fishes have scales, and even reptiles have scales. We have sebaceous glands as well as sweat glands, which are also called as sudoriferous glands. Sweat glands are also called as sudoriferous glands. And in the same time, there are mammary glands and salivary glands, olfactory organs, lens of the eye, a part of the cornea which covers the outer surface of the eye, internal ear and external ear of mammals, including anterior pituitary. So we have discussed about the origin of the pituitary. It is originating from the roof of the buccal cavity, we are saying. From the roof of the buccal cavity means the roof of the buccal cavity is lined internally by ectoderm, and hence it is ectodermal in origin, epidermal ectodermal in origin. In addition to all these, it also forms stomodium and proctodium. So I have already discussed in the earlier classes about this. You might have forgot this. I will be explaining these two again. During gastrulation stage, this is the gastrula, isn't it? So the gastrula has endoderm, which elongates in anteroposterior direction. So here we already have seen that this is the blastopore which becomes anus. That means mouth should be formed on that side, the other side. So when the gastrula elongates and forms a body, this is the body wall, and this is the alimentary canal, and this is the blastopore which became anus, and this is the mouth. This is how it is formed. And during this process, the ectoderm comes inside and lines the interior of the oral cavity. At the same time, it lines the interior of the anal cavity. Okay, and this region of alimentary canal, which is lined internally by ectoderm, is called as stomodium. Stoma means mouth. Okay, stomodium. And this region of the alimentary canal, which is lined internally by ectoderm, is called as proctodium. Thus, stomodium lines, it is nothing but the epithelium of the oral cavity, and proctodium is the posterior part of the alimentary canal. So all these are derived from epidermal ectoderm. Okay, next one, neurectoderm or neural ectoderm. We can call this as, after separation, it forms into neural tube. So we can call this neurectoderm. Previously it is neurectoderm. Now it is a neural tube. And this neural tube gives rise to, number one, brain, spinal cord, and also peripheral nervous system comprising of cranial nerves of the brain and spinal nerves of the spinal cord. So that means central nervous system and peripheral nervous system develop from the neurectoderm. In addition to this, rods and cones of the retina of the eye are also neurectodermal in origin. Why? Because they must be connected to the brain. Unless otherwise they are neurectodermal in origin, they cannot send the impulses, light impulses into the brain, onto the brain. And the next one is optic nerve, skin receptors which are also connected to the brain, auditory receptors present in the ear, and taste receptor present in the tongue, which are also nothing but nervous. All these are set receptor. When you come across with a term like receptor, not the receptors that are present at the surface of the cell, but the receptors that are present in the skin and other parts of the body, which are what we call, uh, which can respond to the stimuli. And these are nothing but nerve endings. Okay, receptors here are nerve endings. When I pinch here, I feel pinching. Why? Because Here we have nerve endings, and these nerve endings we have receptors here. For example, I hit, there is a mechanical receptor or pain receptor that can recognize this, that can respond to this. If I pour some hot water on my skin, there are certain thermal receptors which can respond to the cold and heat stimuli. And these receptors are nothing but nerve endings. Because they are nerve endings, they must be arising from neural ectoderm. And also posterior pituitary. We know that pituitary gland is having anterior pituitary and posterior pituitary. Anterior pituitary is derived from epidermal ectoderm, and posterior pituitary is derived from neuroectoderm. We have discussed already about this while the origin of pituitary gland. Next comes the neural crest cells, the third part of the ectoderm. It gives rise to so many cells. One is sensory neurons, it gives rise to, and satellite cells of the spinal ganglia. Satellite cells are one sort of connective tissue cells of the nervous system. And the cholinergic neurons and adrenergic neurons of sympathetic nervous system. What is cholinergic neurons? The neurons which release acetylcholine as neurotransmitter, and the neurons which release adrenaline as neurotransmitter are respectively called as cholinergic and adrenergic neurons. In addition to this, adrenal medulla is having certain cells which are stained with chromic acid. We have discussed about it. So, when the adrenal gland is stained the with chromic acid, only the medullary portion, the cells within the medulla get pink color because of chromic acid. Hence, they are called as chromatin cells. And these are also derived from neural crest cells. Hence, that is the reason why adrenal medulla, which gives rise to epidermis, epinephrine, and norepinephrine, they are connected to the nervous system. And the C cells of thyroid are those cells which secrete thyrocalcitonin, which is a hypocalcemic hormone. And chromatophores of body, that means whatever the pigmented cells that are present in a body, except those that are present in the retina. And visceral cartilage as well as facial skeletal cartilage. Ciliary muscles of eye, we know in the eye there is lens which is supported by ciliary muscles, and these ciliary muscles and also the wandering cells of the immunity or the blood, that is fibroblasts. All these are derived from neural crest cells. Okay, and after the ectoderm we go to endoderm. Endoderm gives rise to, mainly when you somebody asks in single word, it is alimentary canal. You can tick it, and somewhat going it deeper part where it is going to form other parts in addition to alimentary canal. First of all, alimentary canal means stomach, liver, pancreas, gallbladder, and all the things that are associated with the alimentary canal, even the pancreas. In addition to this, because we know this is mouth leading into buccal cavity, from here there is gullet which leads to esophagus, there is epiglottis which leads to respiratory tract. So because endoderm gives rise to alimentary canal, and the same endoderm gives all rise to the epithelium of larynx, trachea, bronchi, and also alveoli. So alimentary canal, and also from which respiratory system. Primary function is formation of alimentary canal from which respiratory system is derived. In addition to this, it also forms epithelium of urinary bladder, thyroid and parathyroid glands because they are also with the trachea, auditory tube, that means the one which leads from the external ear into the middle ear, and the tympanum, we know that, eustachian tube. The eustachian tube is the one which connects the pharynx with the middle ear, and the prostate and bulbourethral glands of the male reproductive system, and vaginal urethra of the female reproductive system. So these are the main things that are going to be for this is the fate. This is actually the fate of the cells of the endoderm. Okay, and primarily speaking, alimentary canal. And coming to mesoderm. I've just depicted mesoderm is divided into three parts: epimere, mesomere, and hypomere. Epimere is again divided into dermatome, sclerotome, and myotome. So the name itself is clear. Dermatome forms dermis. We have body wall. The body wall has skin. The skin has epidermis and dermis. Epidermis is derived from epidermal ectoderm, and dermis is derived from dermatome of epimere. Likewise, sclerotome, skeletal related, means the vertebral column is derived from the sclerotome. And myotome, myo means muscles. The skeletal muscles and also appendicular skeleton in case of fishes are developing from epimere, which is also called as somatic mesoderm. And mesomere. Mesomere is also called as interstitial mesoderm because it is present in the center. Interstitial mesoderm. And this interstitial mesoderm gives rise to excretory system, gonads, and also adrenal cortex. This one you have to remember. Anyway, you will be remembering this part gives rise to excretory system as well as reproductive system. And we have seen adrenal medulla being developed from neural crest cells, whereas cortex develops from the interstitial mesoderm or mesomere. In case of males, the reproductive system and excretory system are separate. In case of female, reproductive system and excretory system are separate. They have separate openings. Whereas in case of male, both are connected. Hence we call it as urinogenital system. Why we call it as urinogenital system? The male copulatory organ is the only way for excretion and also for the transfer of sperms. So why this is happening so? Because both reproductive system and excretory system are derived from the same region of the mesoderm. And finally, hypomere. Hypomere is divided into two parts again: somatoplure and splanchnoplure. Somatoplure. What is somatoplure? This is gastrula stage with ectoderm, endoderm, and mesoderm. So somatophore refers to these two, this ring. What is that? Ectoderm and somatic mesoderm. And splanchnophore refers to this ring, that is endoderm and splanchnic or inner mesoderm. So here the somatophore gives rise to parietal peritoneum, limb and trunk muscles, linings of the pleura. Pleura is the outer layer of lungs, and also the pericardium, pericardial peritoneum. And the splanchnoplure, that means the one which is present towards the endoderm, gives rise to visceral peritoneum, mesenteron, heart, cardiac muscles, smooth muscles, and also bone marrow. This is how all these three germinal layers are involved in the formation of different parts of the body. And I would like to end this class with a small insight on the extraembryonic membranes. In human beings, let us say when we develop as a fetus in the we develop in the uterus of the mother. So this is the fetus, okay, and this fetus has different layers surrounding it. The one which is near the fetus is called as amnion. This is called as amnion. And this has an amniotic cavity which is filled with an amniotic fluid that protects the baby against the shocks that are impinged on the abdomen of the mother during the pregnancy. And the second one is the one which starts from the gut region, mid gut region. The developing baby has An alimentary canal which starts with mouth and ends with anus. And this is the midgut, this is foregut, and this is hindgut. So from the midgut region, they develops the yolk sac, and thus it has all the yolk that is present here, responsible for directly sending the food to the developing baby. And the thirdly, there is an allantois that is connected to the hindgut region. Allantois is connected to the hindgut region. Okay, and above all these, we have another layer, that is chorion. So thus we have four layers around the developing fetus or the developing embryo. These are called as extraembryonic membranes. They are not in any way concerned with the body of the fetus or the embryo. That these cells are not going to give rise to any part of your body. Rather, they are said to be having varied functions during your embryonic development. We call them also as fetal membranes because they are present around the fetus. Okay, and this chorion forms a connection with the placenta. Sorry, with the wall of the uterus. So when the wall of the uterus it forms a connection, we call it as a placenta. Okay, and if in the formation of placenta chorion takes part, we call it as chorionic placenta. If yolk sac takes part, it is called as yolk sac placenta. If allantois is taking part in the formation of placenta along with the uterine wall, endometrium of the uterus, it is called as allantoic placenta. So here these extraembryonic membranes are also involved in various other functions, and these functions are coming to the amnion first. It develops from the folds of somatochlore, and these are called as amniotic fluids. Somatochlore already I have explained you. And the innermost somatochlore comprises of inner endoderm and outer mesoderm. Its functions are free the embryo from pressures, permitting freedom of motion, and protecting the outer surface of embryo from abrasion. Okay, it frees the embryo from pressures. That means the fluid that is present absorbs the shocks, thus it is protected. And inside the fluid, inside the amnion, the baby is free to move. As different parts are growing on, it is moving, and the movement is also facilitated by this. And it protects. This is amnion, and this is fetus. If amnion is not present against the body of the mother uterus, this fetus skin is exposed and it may get ruptured, abrasion. It may get ruptured, and thus it protects from the abrasion. And coming to the chorion, it is developed from the outermost somatochlore, outer endoderm, and inner mesoderm, and its functions are mainly exchange of substances between embryo and mother because it forms a chorionic placenta. And also, this is the chorion. The chorion forms chorionic villi, okay, and the mother's uterus wall forms crypts. So, the chorionic villi go into the crypts of the mother, forming an intimate connection, and this intimate connection is called as placenta. Okay, and these chorionic villi increase the area of absorption of food material from the mother. And the third one is yolk sac. It develops from outer splanchnic mesoderm and inner endoderm, and it's having two functions. One is storage of food, and during the embryonic development, the formation of blood cells, that is hemopoiesis, is also another function of yolk sacs. And finally, coming to the allantois, it develops from the outer splanchnic mesoderm and inner endoderm, and its main function, when you say this is connected to the hindgut, we are saying to the hindgut it is connected. When it is connected to hindgut, we may think that it is responsible for excretion, but its main function is removal of carbon dioxide. This is what you have to remember. That is its primary function, and removal of urea is its secondary function. Okay, so this is what about the functions of extraembryonic membranes, and the formation of these membranes and other things you'll be knowing when you get into your MSc. So with this, most of the topics, um, those that are not even in the degree syllabus, in view of life science examination, I have covered. Okay, if you want anything other than this in embryology, I will deal with. Otherwise, from tomorrow onwards, as per your wish, I'll start either cell biology part or I will start evolution. Whatever you say, I'll follow.